Great. Well, thank you very much to Norbert for inviting me to give me a chance to present our data here. And the title of my talk today is Single-Cell RNA-seq in the Lung, Effects of Tissue Storage and Dissociation. So in the talk, I will really mainly focus on technical aspects and not talk so much about the biology today. The question we have in mind is, of course, what is the cellular landscape of the human lung, as this is all part of the Human Lung Cell Atlas. And I've already been involved in a couple of studies that are actually published now. There was a paper, Vera Braga et al. In Nature Medicine that came out in 2019. And earlier this year, we've published the tissue stability study with Ilo Madisoon being the first author. And whilst the questions are biological, what I will be doing today is first ask how prolonged cold storage will affect single-cell RNA-seq data quality, and then secondly, what are the effects of the choice of enzymes in dissociation protocols. So our RNA-seq single-cell workflow is probably very similar to that of most people. We start by tissue. Wherever possible, we start with fresh tissue, and we have a very nice collaboration that allows us to retrieve relatively large pieces of tissue from organ donors, but at times we've also used samples from surgery or biopsies. Then, of course, that has to be transported to the lab, where we then digest in order to obtain live cells. And of course, the great advantage of using live cells is that you can sort out those populations that you're most interested in. And then that's followed by sequencing and analysis. Mostly we have used 10x droplet sequencing, but also plate-based full-length sequencing using SmartSeq2. And this protocol at the moment, we're finding that starting with fresh tissue and dissociation still gives us better quality and good depth data. But of course, let me just get rid of that again. But of course, it's also possible to start with frozen tissue where you have much less effect of tissue dissociation because you just have a mechanical disruption of the tissue and then do single nuclear RNA sequencing. And that I will compare some of our data to results with that data, but on the whole, we still find that that gives us lower depth transcriptomes. So it's worth understanding what the effects of the processing are. So I will first talk about the cold storage and then about the tissue dissociation protocols. So for the cold storage and preservation, we've set up the following experiment. We took tissue samples from the lung, the esophagus, and the spleen, as these were supposed to represent 3 different tissues with different sensitivities to ischemia. For each, we did 5 to 6 different individuals. Once we retrieved the tissue, we stored it in hypodermisol, and then stored it, either trying to use it as soon as the tissue arrived in the lab, or stored it for 12, 24, and 72 hours. Now I should point out that our tissue, as I've said, mostly comes from organ donation programs. So the tissue is actually perfused with cold preservation solution prior to being dissected out. Oops, this is going, hang on. This is going totally crazy, apologies for that. So we basically have 4 time points for each donor and then carry out the single-cell RNA sequencing. And here underneath, I've shown some of the QC metrics that already show you, in effect, the answer to our question. And that is there a negative effect of cold preservation of the tissue? And basically the answer is no. I mean, most of the quality assessments that we look at are very stable across the storage time. Here's the number of reads, the number of cells, the median number of genes, and then lastly, we have the number of reads to the transcriptome. So we can see here that for the spleen, at 72 hours storage, there actually begins to be a drop-off in data quality. But for all of the other samples, actually, the stability of the tissue is pretty good. We were very surprised by this. We thought there would be a much earlier drop-off of quality, but of course, logistically, it's very, very helpful that it does appear to be possible to store tissue. But of course, the tissue itself does actually deteriorate as we have shown from tunnel staining in tissue sections. Here are a couple of different tissues, always at time 0 and time 72. And as you can see by the brown staining, there is an increase in apoptosis at 72 hours in all the different tissues that we looked at. But it appears that the tissue dissociation and the subsequent removal of dead cells really gets rid of that effect. I mentioned that there was a decrease in the data quality in the spleen, so it's maybe quite interesting just to dig into that a little bit more and understand what's going on. And one effect that I thought was quite interesting is actually that the ratio of exonic to intronic reads changes as the quality of the sample decreases. So here in the spleen, we get fewer exonic reads, but more intronic reads, and we interpret that to mean that RNA in the nucleus is probably more stable than RNA in the cytoplasm. And this is just to show that that effect was actually seen in all the individuals we analyzed. But now coming actually back to the data in the lung, because I'm here as part of the lung seed network, we sequenced approximately 60,000 cells and actually obtained a good representation of all the cell types that we expected to see. The particular association protocol we used is very heavily enriched for immune cells, which was actually intentional. And then looking here on the right-hand panel at all the individual samples, so with the data being split up both by individual donor and then also by time point. And the first thing that you can see is that there is a lot of sample-to-sample variation overall, but also that the sample variation within a donor is less than that between two different donors. And in particular here, you can see this donor has a lot of these yellow cells, which represent mast cells, whereas maybe this donor here, 356C, has a lot of NK cells. So clearly, the immune environment in individuals' lungs can be very, very different. And I think this is an important observation, as I think it will inform the number of individuals we will probably have to analyze in order to get a good human cell atlas. Another aspect that was quite interesting was the heterogeneity of the samples in terms of the microanatomy. So here are just two lung samples, and mostly we saw very few blood vessels. But in these samples that I've highlighted here, there were more blood vessel cells. And we think this is most likely simply because the microdissected piece of tissue simply had more blood vessels in it. But so when you're starting with human tissues, this heterogeneity has to be taken into account. And here's another examples in the esophagus where there was actually only one donor where we saw representation of mucosal glands. And indeed, we could also see those glands when we looked at histology sections from those individuals. We then asked whether the cell types that we retrieved change with storage time. And we found that over 24 hours, actually for none of the tissues we looked at, there was no significant change, although clearly here there is a trend for a reduction in T cells that became significant by 72 hours. In spleen, B cells increased, but this was not statistically significant. So certainly within 24 hours, we believe that we get good representation of all the cell types that we can detect. We also wanted to ask what the effect of the dissociation and the storage was per se. So we compared our single cell data to RNA bulk data by taking bulk data and then making pseudo bulk data from the single cell. And here's a PCA that either shows the method, the donor, the time of storage, or the organ it came from. And as you can clearly see, the samples segregate by method as well as by donor, but really the time points are widely dispersed so that that is not a primary determining factor of variance. We also looked at the gene signature itself and actually compared it to the dissociation signature that Van Oudenaarde first described, and actually we do find that it's quite similar. We also looked at the changes with extended storage time, so comparing T0 with T24. And again, this overlapped quite significantly with the stress signature. So we interpret this as likely to mean that the cell probably has one particular stress pathway it can activate, and it won't vary that much depending on exactly what the stress is that the cell is under. But then, so for this part of my talk, the conclusion is really that you can obtain good single-cell RNA-seq data after 24 hours of cold storage. And the data we have generated is all available in databases. Here, the Tissue Stability Cell Atlas.org for the Madison et al. Paper, and also the Lung Cell Atlas.org for the earlier Nature Medicine paper. But now I want to come on to the effect of enzymes in tissue dissociation protocols. Now, I have to do a slight disclaimer here. We did not set up this experiment as a technical experiment. Rather, we carried out a number of different biological studies, and now that we have a reasonable amount of data, we're trying to query that data to get some idea of what the effects of different dissociation protocols are. So the initial data I showed you in the stability study was just for parenchyma, but we also wanted to look at the trachea and bronchi, which are much tougher. So we used a slightly different protocol where we first used liberase TL to dissociate the tissue, and that fraction of single cells was then split into CD45 positive and negative cells, but tissue that was not completely digested, so the remaining debris was exposed to trypsin, and then all cells were loaded. And so you can see here in the UMAP that these are the CD45 positive cells. They were clearly enriched after a CD45 column, although the enrichment is clearly not perfect. There are a representation of a number of different clusters in the CD45 negative population. But very interestingly, once we also treated with trypsin, some areas were really much better represented in these cell populations. And in particular, we see much better representation of stromal cells, but also basal and goblet cells seem to be better represented after trypsin treatment. We also noted that most of the trypsin-released cells in this experimental setup were not CD45 positive. So as we went forward for a larger experiment, we actually combined the CD45 negative fraction with the trypsin fraction to reduce the number of experiments. So the study we had set up for the biological question was actually to look at different areas of the lungs. So we wanted to look at the trachea, the primary bronchi, the sort of lower airways, and then the lower left and top left parenchyma. And we carried this out in 5 individuals. So that means we now actually have quite a few data sets that we can compare. The first published study in Nature Medicine used disphase, collagenase, and elastase. Madison et al. Used collagenase, and in the dissociation protocol where I showed you the UMAP, we used the liberase plus trypsin. Then the 5 location uses the same enzyme, but actually uses a different sequencing protocol. All of these single-cell transcriptomes were mapped with the same computational pipeline, and then we removed ambient RNA with SUBEX. And then we actually used the removed reads, the SUBEX, to compare what the effect was on the transcriptomes, because if particular cell types die, then you get a different representation of SUB in your sample. This is now a representation of all the different studies. The first study in Nature Medicine still had quite a large amount of contamination, up to sort of 12–15%. That got better in the subsequent studies, down to about less than 5%. And in the most recent study, we are probably down to an average of 2.5% or so. But what's most noticeable is that there's really a lot of variation between individual samples. And that is even the case if individual samples come from the same organ donor, as indicated by the colors here. Interestingly, when we looked at the locations where the samples came from, there didn't seem to be any particular patterns. So the origin, the sort of spatial origin of the sample didn't seem to have a big effect on the SUB. So we then used all of those samples and clustered the reads that were removed by the SUPEX filter. And the most striking thing here is that it clusters primarily by the RNA sequencing protocol. This was 3' 10x and this was 5' 10x. But within these studies, this was the 5-location study, and these were the Braga and the tissue stability studies, there were actually different protocols used. So here we used liberase and then liberase and trypsin, and here collagenase and collagenase D. And yet, despite these different protocols, the samples really cluster by donor rather than by dissociation protocol. So while this is not a very quantitative analysis, it does, I think, indicate that different dissociation protocols actually add less variation than might be expected. But these are, of course, all the reads that we're actually throwing away. And what we would like to know is what happens to the reads that are still in the data. So in order to do this, we did an analysis to understand the variance components using a linear mixed model. And it was done for all the different cell types individually. And here I've just picked 3 different examples. So we feed as variables the donor, the batch, the count, the study, which is effectively the sequencing chemistry, and then also some medical metadata like smoking, BMI, and age, and so forth, and also the dissociation protocol. And actually, when we compare all those metadata variables, we find that the dissociation protocol is one of the least contributing factors. And things like the cell type itself and the donor give much, much bigger variance than the protocol itself. So I think in the overall scheme of things, that's actually really good news, because it should hopefully mean that going forward, it will be reasonably possible to integrate different data sets, even if they've used different dissociation protocols. And this data was actually done for parenchyma samples only, for 17 different donors. And this is another view at a subset of this data, where I just wanted to highlight again how much, whilst the actual cell type transcriptome doesn't change, the proportion of cells released by different protocols varies enormously. So this was all from the same donor, A26, but the first protocol uses collagenase, which releases preferentially immune cells, as you can see by all these different colors here. These were a mixture, these were liberase and then liberase and trypsin, where you can see that there's an increase in stromal and mesenchymal cells that are being released. And we then also compared that to a nuclear sequencing protocol, which released far more AT1 and AT2 cells, which are the very fragile cells in the alveolar of the lung, which we know are often destroyed by enzymatic digestion. So we know that nuclear gives actually a much better representation of the cell types as they were in the tissue. But as I said initially, on the whole, we still find that the data quality of this tends to be less good than that of the fresh cells. Here's another comparison of single cells versus nuclear, but this time, rather than looking at the parenchyma, we're looking at the trachea. This is a UMAP from the 5-location studies, and I just want to point out 1 or 2 little observations. So for example, here, there was a very small population of chondrocytes that we identified, which now in the trachea sample, in the nuclear samples, are much, much more prominent. And that really makes a lot of sense, because the chondrocytes sit in the cartilage, and the enzyme digestion is just not harsh enough to actually release those cells. We also find other interesting things, like here, for example, we have a number of different muscle clusters that did cluster into separate groups. But when we look at the nuclear data, we actually find that the different clusters separate very nicely. So you can also see that different approaches to sequences can have very different effects for different cell types. And here, clearly, for the muscle cell types, the single nuclear sequencing gives much cleaner data, much better resolved data. But so that's really as far as I want to take you with the technical study. just a brief look to the future. Of course, we generated this data set to look at the different locations and understand how cell type transcriptomes vary with location. And here I've indicated the same UMAP with the cell types with different locations. And I think there are some interesting indications. For example, here in the myeloid compartment, you can see that they are really not evenly distributed by location, and they're distinct areas for the trachea, for the parenchyma, and for the bronchi. So we're now digging into this data deeper and really trying to understand how cell types vary with location. And I think that brings me to the end of the talk. Just to relist my conclusions. Our cold preservation study showed that you can get good quality RNA-seq data after cold storage, certainly up to 24 hours, but actually in many cases as far as up to 72 hours. But bear in mind that this is with cold perfused organs. We also looked at the cold preservation gene signature, and that overlaps with the stress signature due to dissociations. I showed you that different enzymes were released, very different cell types, but also that once the cells are released, they do seem to have relatively similar stress signatures. And then lastly, this conclusion I think is very obvious to anybody who's worked in the field. At the end of the day, each tissue will really have to have its own optimized protocol, and it will depend very strongly on the exact scientific question that you want to ask. In our lab, we're getting fonder of single nuclear sequencing because really the improvement of the sequencing chemistry from to the 3' version 3 has meant that now the single nuclear data is becoming of better quality. So with that, I'd like to stop and acknowledge all the people who've been involved. I'd like to thank Sarah Teichmann, Mike Stubbington, and Ollie Stegel, who were also involved in the tissue stability study. The practical work for that was primarily done by Elo and Anna, and here are many other people from our lab who've also helped. I'd like to say a big thank you to Kourosh Sarag-Parsi from CBTM, who provides us with all the human clinical material. These are collaborated from the University of Cambridge and from Sanger Pipelines. And then I'd like to acknowledge the funders. The Chan Zuckerberg Initiative funded the tissue stability study, and Wellcome the 5 location studies. And last but not least, I'd like to thank the donors and their families for allowing us to use their tissue. Thank you.
Thank you, Kerstin. Does anybody have any questions? I realized I said use the raise hand function, but there is no raise hand function currently. Oh, yeah, go for it.
Hi, my name is Nils Lindström. I'm from USC. That was a really lovely presentation. I was wondering if you've seen in your nuclear sequencing, if you've seen any of these artifacts, so-called, about intronic, genes rich for intronic, large introns being captured and overrepresented in your data.
I don't think we've specifically looked for those large transcripts, so I'm not sure that we've noticed them. At this stage, we've primarily just tried to take the data and annotate the cell type and look at proportions. So, but that's an interesting thing. We might look at that and see whether we can see evidence of that.
Or, go ahead.
Oh, hi, Kerstin. That was a nice talk. Just to add to that last question, we have seen that genes with larger introns are more represented in single nucleus data, probably because there's just more poly A stretches in the introns. But yeah, great talk. I would just add that I think I agree with you that every tissue needs its own processing, but also its own sort of computational pipeline. So when you were talking about the single cell versus single nucleus for your lung data, I noticed you had way fewer immune cells in the nucleus data, and that's something we absolutely see as well. And so I was wondering, did you change like any of the cutoffs you were using when analyzing the single nucleus data? Because we have found that T cells, they just have way, they're much smaller and the nuclei are much smaller, so we have to really lower our cutoffs in terms of like the complexity to detect them. So just curious if you had played around with that?
Yes, so we have in various data sets, we've absolutely seen the same thing, that at times we had done the normal cutoff as sort of recommended by Cell Ranger and found that there are practically no T cells. And then when we manually reduced the threshold, suddenly we saw quite a lot of T cells. But I think for the data I showed you, the cutoffs were all, no, actually they weren't the same. They were actually sample specific. So that it was actually quite manual. I think at the bottom of the slide, I said that they all had the same alignment and the same soup X, but thresholds were done on a sample specific basis. And then we combined the data using BBK and N. But I think, I mean, in our lab, we've actually looked at lots of different tissues. And I think it's fair to say that because immune cells are used to, in a way, being able to go out on their own and go into the bloodstream, they are actually quite okay with being digested and being in a single cell state, whereas many of the other cell types are much, much more sensitive. So most enzyme digestion protocols, especially when they're a bit more harsh, really favor immune cells because you're just killing other cells. And I think the relatively small number of immune cells in the nuclear protocol is probably a real representation of the fact that in a normal tissue, immune cells aren't that abundant. It might be different if you're looking at clinically diseased samples where you have higher immune infiltrates, but that would be my judgment on that.
Thanks.
Shyam, do you want to ask your question?
Yeah, hi, this is Shyam Prabhakar from Singapore. Yeah, very nice to see this study. And my question is kind of similar to the previous questioner because we had done informally something similar between my group and another group, looking at the same tumor samples, two different dissociation protocols. And then sometimes we use the same protocol. And we couldn't really agree between the two groups on how the protocol affected the cell type abundances because their conclusions are different from ours. And then we finally realized that it's because they were using different QC cutoffs from what we were using. And so I think it's, you know, at least in our experience, it was not that simple as this dissociation protocol gives you this cell type profile, that protocol gives you that cell type profile. It was more that this combination of dissociation and QC cutoffs gives you this profile. And the QC cutoffs were almost as important as the protocol. So it's kind of a confusing picture. I'd be curious to know, manage to make sense of it and disentangle those two.
I mean, as I said, the study for the enzymes wasn't designed as a sort of technical study. We just looked through the data that we have. So it is actually quite difficult to disentangle the effects because we have organ donors. And of course, then the biological, and you know, your organ donor was involved in a particular study. And then the biological effect is always confounded with the technical effect. There was, I think, only that one donor, A26, which we had, where we looked at different protocols within one donor. It also, these studies get very expensive very quickly if you want to do multiple donors with multiple enzymes. So it's often a little frustrating because you're following a gut instinct rather than, you know, hard and fast statistically significant data. And we also find that if we, like in the tissue stability study, where we're actually reasonably confident that, you know, it was the same person doing the same protocol just 12 hours apart, we still see a lot of sample to sample variation. And we don't really know where that comes from, other than that, you know, the person became a little bit more tired, or it wasn't morning, but lunchtime, and it was hotter in the lab, or whatever it is. But all those tissue handling steps that are not just the protocol, but also how long it waits in between, and whether there was a phone call and they were sitting around on ice or whatever it is, it all adds quite a lot of variation. And I think one of the overriding messages is also that this data is very noisy. And so, but nevertheless, I was personally surprised that when we combined all the SUBEX data, that actually we saw so little effect of enzyme dissociation on the clustering. that that all the other factors were more overriding, like the chemistry. And I would agree that the analysis pipeline would also be one of the factors that adds quite a lot of variance.
Thank you very much, Kerstin. With that, we're going to move on to Steve Henikoff and Anoop Patel. Steve, if you'd share your screen. You know what we're missing here? Applause. I may need to find an applause sound effects for the end of each of these talks. It's all yours, Steve.
How's that?
Great. Looks
okay.
good.
So thanks, Norbert. What I'd like to do is bring you up to date on what we've done with our cut and tag method that I described last year at the kickoff meeting. And we're going to do this in two parts. First part, I'll describe our a streamlined version of cut and tag we call cut and tag direct, and also how we've automated it. And then I'll turn it over to Anoop, who will talk about single cell cut and tag for the 10X platform that is applied to heterogeneous tissues. Okay, by way of introduction, just let me point out that the enzyme tethering method of cut and tag, like cut and run, is fundamentally different from ChIP-seq in the following ways. ChIP-seq that you're familiar with, you start out by cross-linking and solubilizing, whereas with cut and run, for example, we just permeabilize cells and the cells remain intact throughout the entire protocol. So with ChIP-seq, you would then add a specific antibody to the solution, but here we're allowing the antibody to diffuse into the permeabilized cells. So it will say find its target transcription factor, for example, and then we add protein A emanase, a fusion protein, where the protein A part will stick to the antibody and tether the emanase, such that when we activate the enzyme in the case of emanase with calcium and it cleaves on both sides, we release the fragment into the supernatant where we extract it, and then the rest of it is similar to what you do for ChIP-seq. Now, with cut and tag, and this is work that was initiated by Hatice Kayagakure when she was a postdoc in the lab, we substitute protein A TN5 for protein A emanase. Now, TN5 is a cut and paste transposase that you may be familiar with because it's the basis for the Illumina and Nextera system and also for the popular TAG-seq method, but here we're tethering. So when we tether and then add magnesium, when we bind and then add magnesium to activate the enzyme, it'll drop its payload of adapters, and then it's simply a matter of extracting DNA and amplifying the library for sequencing. And this takes 1 day from live cells to purified libraries. Now, the new part, cut and tag direct, is we've gotten rid of this extraction, DNA extraction step, where we add SDS, proteinase K, then do phenol chloroform extraction, ethanol precipitation. We don't do that for cut and tag direct because we just add a little bit of SDS and heat it up in order to remove the enzyme from the DNA. And then we neutralize the SDS with non-ionic detergent, in this case, Triton X, and then that means that we can just do PCR directly. And the whole thing is done in one tube from cells, or in this case, nuclei, to purified libraries just by tube transfer. And because of the simplicity of it, when the COVID pandemic hit and we had to shut down, I realized that, well, there's no toxic materials, there's nothing dangerous here. And it just takes a little bit of equipment in order to run the whole thing. So I brought some of the materials home, magnets and the PCR machine, and put it on a counter in my laundry room, which is just a few steps away from where I'm sitting right now, and started trying. It turned out it worked actually quite well. And we then put up a bioRxiv manuscript describing the kinds of results that you get using this sort of stripped down simple procedure. So this shows some of the data in the bioRxiv manuscript. What you see is for cut and tag at home, we call it, that we do H3K27 trimethylation to map polycomb silence domains. And this is a low cell number test where we've gone down to from 60,000 cells down to 60 cells. And you can see whether this is carried out in the home or in the laboratory or extraction. We get pretty much a very similar nice clean results. Similarly for H3K4 trimethylation, which is a mark of promoters, say a little bit more about, even down to 60 cells, we get pretty good results. Same as at home and in the lab for the human K562 cells. Now, because of the simplicity of the method, we also, Derek Jansen's postdoc in the lab has also modified it for automation, following pretty much the same procedure that he introduced for auto cut and run. And this shows for auto cut and run where we've disaggregated tumor samples. It can be done in cells or tumor samples or nuclei, and mixed with magnetic beads arrayed in 96-well format, and then ran the whole thing on the robot. And what Derek's been able to do is to do auto cut and tag, and that has a few advantages over auto cut and run. First of all, it's faster. It takes a day rather than 2 days for the whole thing. And because the signal noise is a little bit better, we've gotten accustomed to basically doing 96 samples on a single two-lane flow cell. So it's actually it's not only quite efficient and fast, it's also low cost to get high quality data. And we're currently, when things calm down, we expect to start to use this for patient samples with our collaborators. This is set up at the Fred Hutch as a shared use facility. So one other point I'd like to make is that we started to use lightly cross-linked cells for a few reasons. So the light cross-linking that we do came from a protocol from another laboratory that they published, in which they used 0.1% formaldehyde for 2 minutes. And that turns out to be enough in order to prevent the kinds of clumping that can be a problem both with cells and nuclei. And so that makes it particularly suitable for a single cell application. So this just shows the kind of quality that we get. We use the fraction of reads in peaks, the FRIT measure that was introduced by the ENCODE project in order to measure data quality. And you can see that the fraction of reads in peaks it levels off just above 0.80%. And we get that with only 500,000 reads, which is quite good compared to other kinds of methods. And this shows, there are actually 8 curves here. It shows for replicates, different biological replicates, for native cells, cross-link cells, native nuclei, or we cross-link the nuclei directly, we get pretty much the same results. And although we didn't sequence very deeply here, if we pool them, you can see that we maintain the signal and noise advantage over ChIP-seq from the ENCODE project for even going out to here 18 million reads. So there's a lot to recommend this for chromatin profiling in general. As I mentioned, we wrote something and put it up in bioRxiv. At the same time, we put the new protocol up on protocols.io, and they're linked to one another. So it's easy to look at the protocol and then get some background to see what the results are and how you can actually do it at home. And actually quickly, some people actually got it to work for them. Okay, what I'd like to close with is to point out the richness of the human epigenome, so to speak, that we can see by just using different antibodies. In this case, 5 different antibodies for 4 positions on the histone H3 tail. In addition to the ones I mentioned, H3K27 trimethylation, which gives us polycomb developmentally silenced domains, and H3K4 trimethylation, which gives us promoters. We also look at some other marks. So let me just explain. If we just blow up this region here, it's a representative region of the genome. What you can see if we use H3K4 dimethylation, we see that in addition to the promoter peak, we also get a peak which, using a massively parallel reporter assay, this from Bos-Van cell for K562 cells. This is identified by function of promoter enhancer. So we can actually pick up this nearby enhancer just downstream. And this is confirmed by, you can see the ATAC-seq peaks actually are in the accessible region between these nucleosomes. And finally, I just mentioned that in addition to H3K27 trimethylation silence domains, when we use H3K9 trimethylation, we pick up these peaks, which turn out to correspond for the most part to endogenous retroviral elements. Now, and this one is in the ERV1 family, but these ERVs, there are 600,000 ERVs in the human genome, covering about 8% of the entire genome. So it's a good part of the genome that we can identify just using this. And this is an unexplored area that I think it'd be worthwhile to look at more with respect to like developmental changes as retroviruses get silenced. Okay, with that, I'd just like to acknowledge our Fred Hutch Seed Network, Kami Ahmed, Rafael Guittardo, and Derek Janssens, Mike Mears, and you'll be hearing next from Anoop, who will tell you about what he's been doing together with Scott Ferlin and Anka Mihalis. So I will stop sharing and turn it over to Anoop.
All right, thanks, Steve. It's great to talk here with the group. So I'm actually going to be presenting just our very preliminary work using tethered transposases to perform single-cell chromatin profiling on the 10x single-cell ATAC-seq platform. So, hang on a sec. Okay, so the focus of my lab as a practicing neurosurgeon, we're very interested particularly in a highly malignant brain tumor called glioblastoma. And this is just an example of a patient that I'm going to kind of use as a case study for the rest of the talk. So this is a patient that we operated on back in March of last year. We removed the tumor. We then go through and we take the tissue back to the lab. We generate a variety of patient-specific avatars, including cell lines and patient-derived xenografts. And then we put the tissue through a variety of single-cell sequencing applications to try to understand a little bit more about the disease. And then, the patient goes on to undergo treatment, in this case, radiation therapy. And then, unfortunately, 5 to 6 months later, the patient succumbed to their disease. And one of the things that which is actually not atypical for this disease, it gives you a sense of really how aggressive it is. And so one of the things that we're really trying to understand is why does this disease always come back, regardless of our best efforts at surgery, chemotherapy, and radiation. And so in order to get a better sense of that, we've partnered with our neuropathology colleagues to develop a rapid autopsy platform where we can get tissue at a relatively short postmortem interval, in this case, less than 8 hours. And what we're trying to do is actually use non-genetic sources of heterogeneity to try to understand this disease process, both at the outset of the disease and then at the time of recurrence or at the end of the disease process. And so our group and other groups have done a lot of single-cell transcriptome work in glioblastoma in the past. We've decided that we wanted to expand that. We've switched over to doing single nucleus RNA sequencing for a variety of reasons, some of which were discussed in the previous talk, mainly due to the fact that we actually get a much better representation, we feel, of the cells present in the microenvironment. And so that's one thing that we've been doing. Then the other thing that we've been really focusing on is trying to use other orthogonal methodologies like single-cell ATAC sequencing to try to understand the regulatory networks that might underlie this disease, as well as its progression and ultimate, what the disease looks like at the end stage. And so I had showed Steve some of this data a while back, and he had mentioned that he was developing single-cell CUT&Tag as a way of doing histone mark-specific chromatin profiling. And so we were particularly interested in K27 trimethylation based on some of the previous work that we had done. In this case, we're looking at an in vitro model of glioblastoma where we treat the cells, in this case with a receptor tyrosine kinase inhibitor, and we actually show that there's widespread chromatin remodeling and changes in H3K27 trimethylation at regions of facultative heterochromatin that are then de-repressed and result in the emergence of a cell state that kind of is resistant to treatment. And so we wanted to really see if we could use this methodology to try to understand what happens when the tumors ultimately grow and then recur. So most of the rest of the talk is really about less about the biology and more about the technical aspects of getting this to work, which I think is of a little bit more interest to this particular group. So we basically took the protocol that Hatice had developed in Steve's lab, and Anka Mahalas from my lab worked closely with her just to basically make some very simple modifications to the regular workflow to get this to work. So we switched out the TN5 to a protein A tethered TN5, and then we pre-incubated the nuclei with a validated K27 trimethyl antibody overnight. And then we just ran it through the standard 10x ATAC-seq protocol. And actually then ultimately also used CellRanger to do sort of cell barcode identification. And what we got out the back end was not a total surprise. CellRanger didn't really know what to do with this type of data. And a quick shout out to Scott Furlan, who's a hematologist and computational biologist at the Hutch, who really helped us kind of look under the hood of all of this and really develop some of the computational methods to better analyze this data. So a lot of what I show has been done in close collaboration with him. So basically what we found is that CellRanger didn't do a very good job, mainly because the algorithm itself focuses on the idea of calling peaks within the aggregate data and then using QC metrics based on that, things like fraction of reads overlapping peaks, to decide what's a good quality cell and what's not. And so CellRanger 1 did a pretty poor job. CellRanger 1.1 actually did a slightly better job, but we were still getting relatively low metrics for things like fragments overlapping peaks. And if you delve a little bit deeper, that makes a lot of sense just based on the way that the algorithm works. So it was designed for single cell chromatin accessibility data, which of course is peaked. And as Steve showed, K27 trimethylation data is really these sort of broadly repressed regions of facultative heterochromatin. And so here I'm just illustrating on the aggregate K27 tracks and then showing the peak calls that were done by the 10x CellRanger output. And so you can imagine if we're only counting the reads that are contained within these small sort of narrow peaks, we're actually throwing out a lot of the really useful data that we could be gleaning from these libraries. And so Scott and I decided that we were going to take advantage of a new methodology for analysis of single cell chromatin accessibility data that recently been published by the Greenleaf Lab. This is their platform called ArchR or Archer. And so the basic workflow here is that they recommended 500 base pair genome tiles. We used 5 kilobase genome tiles, mostly because we selected that as approximately half the size of a polycomb domain, somewhat arbitrarily, but I'll just show the data for that. And so we did 5 kb genome bins across the genome, and using the fragments file, quantified the fragments in those genome bins. We then integrated the data for the primary tumor and the autopsy specimen. We filtered for doublets. We did some batch correction using the Harmony algorithm. And then we used really their kind of off-the-shelf iterative LSI for dimensional reduction, and then Leuven clustering, again, just sort of whatever the standard package sort of does for us. And we then, did a bunch of QC metrics. I won't show them all here, but this is just to give an example. So on the left is from the same sample, the single nucleus ATAC-seq, just showing the fragment size distribution. You can see good nucleosomal periodicity there. On the right is the same fragment size distribution for our single cell CUT&Tag libraries. And you can see again, the overall fragment length distribution was larger, but you do see nucleosomal periodicity. So it gave us some confidence that we were actually looking at something that was somewhat meaningful in terms of chromatin data. And so then, just looking at sort of what the workflow gave us, this is what the low dimensional embeddings of the data look like. You can see that there is fairly robust separation between the clusters within the data. We then, if you look at the data on a per sample basis, you can see that there are some specimen-specific clusters, but then there's also clusters that seem to overlap, which makes sense and suggests that our batch correction algorithm did actually function to some degree or another. And so then we sort of turned to the issue of trying to figure out what each of these clusters in the single cell cut and tag data actually represent. And so because we have RNA-seq and ATAC-seq data from this same patient specimen, we do have some a priori knowledge of kind of what to expect on some level. But cluster annotation is kind of an issue, because if you think about what K27 trimethylation data is really telling us, we're sort of looking at regions where signal correlates with a lack of activity or a lack of expression. And quite frankly, there's just none of the current single cell analysis packages are really written to do that. For the most part, they're all written to sort of use positive enrichments as a way of identifying marker genes in subpopulations. And so we actually took a couple of different approaches to this, which I'll kind of work through here. So the first approach that we took was taking advantage of the concept of a gene activity score. And so the way ArchR does this is it defines the gene activity score as accessibility within the gene body, and then uses an exponential weighting function that accounts for the activity of distal regulatory elements, in this case in a distance dependent fashion, and then also imposes some gene boundaries so you don't get kind of overlap of regulatory elements that are actually related to a different gene. And that spits out sort of an imputed gene activity score for a particular region. And then the ArchR workflow implements this methodology called MAGIC, which actually smooths these gene scores across cells locally in the graph in order to kind of clear up the data and make it a little bit more apparent what's going on. And so with this workflow, we would expect that activity at a marker gene for a particular cluster would suggest that it was actually not that type of cell. And so this actually worked fairly well for some of the clusters. And so here's just an example of a couple of these. So PTPRC, the PTPRC locus, which is more commonly known as CD45, which would be in our case a marker of microglia. You can see that this method clearly identifies a cluster of low activity, low polycomb activity at this particular locus, where the remaining clusters actually do have some relative moderate to high activity at this locus, suggesting that this cluster of low activity represents microglia. The same thing is true for the RBFOX3 locus, more commonly known as NUN, which is a marker of neurons. And again, as you can see here, there's clearly a cluster that separates out as having very low polycomb activity at this locus, suggesting that this would in fact be neurons. And so we then thought about, as a second way of doing this, we wanted to think about ways that we could leverage pre-existing bulk data to try to get some idea of what the clusters actually were. And so to do this, we took advantage of a really nice feature in the ArchR package, which allows you to subsample bulk chromatin accessibility data, or in our case, ChIP-seq data, and or cut and run data, and essentially develop a pseudo single cells from that bulk data, and then project those pseudo single cells onto the actual single cell data. And so Jay Sarthy from Steve's lab was kind enough to do some bulk cut and run for K27 trimethylation on an actual stem cell line that we isolated from this same patient. And so presumably that stem cell line contains only tumor cells, and so would give us a good sense of what the actual K27 trimethylation profile of the tumor cells should look like. We then kind of bolstered this with ENCODE data for monocytes, which was a kind of our proxy for microglia, since there wasn't a good data set for that, and then a couple of other ones. We generated pseudo single cells by subsampling the data, and then we projected this onto our single cell data. And this actually works pretty well. And so you can see this is the, in pink is the UW7 glioma stem cell line, subsampled and then projected onto the data. And you can see that it actually identifies a couple of clusters here as being very, very similar to the K27 trimethylation profile of the cell line. And then using the ENCODE data, actually it very readily identified this cluster as monocytes. And this actually was very much in line with our previous data where we used the sort of gene activity scores. So kind of combining these two methods and doing sort of a little bit of manual curation along the way, we actually were able to develop some basic cluster annotations that are kind of shown here. And so we're currently working on some additional methods to do this, but really the next steps are that we sort of wanted to focus on trying to understand what are the loci that are actually different across these clusters. And so we're still in the process of doing a lot of this analysis, but I thought I would just show basically some pseudo bulk data from aggregated cells within a cluster to kind of give you a sense of what the data actually looks like. And while it is quite noisy, you can actually clearly see that at certain genome loci, there is a preferential polycomb activity within different differently across the different clusters. And this is another example of a locus where there is polycomb activity at this gene Sec61G in a subset of the clusters, but clearly not in the others. And then just here's, yet again, another example. And so there are a variety of next steps that we're kind of looking to do. We're trying to work on developing better annotations. We've used in the past this infer CNV platform, which basically uses background noise in single cell data to essentially infer copy number changes, and that allows us to positively identify tumor cells. And we have some preliminary data to suggest that that's actually going to work. The other thing that we've looked at or thought about doing is looking at an anti-correlation with a TAC-seq or H3K4ME3 profiles. And Scott Furlan's been thinking about training a neural network model to do this for us. And then the holy grail of this would be to be able to combine this type of profiling using some sort of a multi-seq method or a site-seq type approach in order to get either transcriptome or surface marker information. Of course, we'd like to expand to additional marks, including some other activating marks, as well as some other marks of heterochromatin, as Steve had showed. And of course, also would be very interesting to try this with transcription factors. We also, of course, want to expand to more samples. As Scott has a particular interest in blood and leukemia. Jay has an interest in pediatric glioblastoma. And then one of the things that became apparent from our attempts to use the ENCODE data sets is that there's really not great data for brain for K27 trimethylation. And so it would obviously be very interesting to do normal brain and sort of kind of an atlasing effort to develop K27 trimethyl or other histone-specific mark profiles for normal cell types as well. And so with that, I'd really like to acknowledge Anka and the other folks from my lab who contributed to this effort, and then really this very close collaboration with Steve and the folks in Steve's lab, Hatice and Jay, and then of course Scott, who without which a lot of this analysis would have really been impossible. So that's pretty much all I've got, and I guess we can take questions.
Thanks, Anoop. Any questions? If you could raise your hand and then unmute. No? I think we're at time, so we're losing a lot of people anyway. But thank you guys for your presentations. Like I said, these recordings will be made available online, and the next seminar coming up, we're going to have Catherine Schmidt from Matthias Henig's group, and then Koresh Parsi, I apologize for that, from the Cambridge Biomedical Biorepository. It's on your calendars. Share the link with your friends and colleagues, and we'll see you next week, or in 1 month rather. So thank you all for coming. Have a good day.